Hi, so this recording will deal with Lecture 4, and we're going to be reviewing genetic principles that are relevant to blood banking. So for our objectives, firstly, we're going to define the term blood group system with regard to genetics. We will differentiate phenotype from genotype. We will define a few terms, including gene, allele, haplotype, and polymorphic. And we will distinguish homozygous from heterozygous and provide examples using blood group system alleles. Moving on to objectives number 2, we will define the dosage effect and explain its significance in testing. Explain the difference between cis and trans and their effects on gene interactions. Define codominant inheritance. And explain phenotype frequency and how it can be used to find compatible red blood cell units for a patient in need of transfusion. So let's begin with our blood group systems. Blood group systems are simply groups of antigens that are on a red cell membrane that share related serologic properties and genetic patterns of inheritance. So the first blood group system, for instance, that we will be studying is the ABO system. And then from the ABO blood group system, we'll move on to the RH blood group system. And so this just gives you an example of some, that you may have heard about already. When we get into blood group genetics, this is just a basic review, which you probably have had previously in a biology class. Genetic material is located in DNA, and DNA is contained in chromosomes, and chromosomes happen to be in the nucleus of every cell. Genetic material is replicated either by mitosis if we have a somatic cell, or meiosis if it's gametes. When we move on to looking more carefully at genes, genes are the basic unit of inheritance on a chromosome. The locus is the site at which a gene is located on a chromosome, and I like this word because think location, and locus, basically you can figure out what it means. Alleles are alternative forms of a gene. So they are opposite antigens that are produced by alleles at the same locus, and they're antithetical. So a little bit later, we will look at the KEL blood group system, and within that system there is a KPA antigen, which happens to be antithetical to a KPB antigen. And so that's an example I can give you now. Multiple alleles that are at a single locus are considered to be polymorphic, many, right? And so our ABO system, which we will be studying first, is a good example of polymorphism. So here we see a set of chromosomes, and we see gene locus. And when we look at each individual chromosome, we can see that there are two different alleles. The chromosome on the left has an A allele, and the chromosome on the right has a B allele. So this is polymorphism. With blood banking, it's important to differentiate between phenotype and genotype. We'll be talking a little bit about this in Lab 1 as well. So when we talk about phenotype, these are physically observed expression of traits that we may see in some of our testing. The phenotype with blood banking is determined by the agglutination of red cell antigens when we use known antisera. Okay, so it's phenotype. It's a physical expression of a trait. So for example, a person who shows no agglutination when their red cells are added to anti-A or anti-B antisera would have a certain blood type. Okay, so if they don't agglutinate with antibody against A, it means they lack the A antigen. And likewise, if they don't agglutinate with antibody directed against B, they also lack the B antigen. So if we were in class, I would make you guys answer this for me. Basically, this means they're type O. Okay, so O basically lacks both of those antigens, and that, in this case, would be a phenotype based on our serologic testing using antibodies. When we get into genotype, this is the actual genetic makeup meaning the alleles that an individual actually inherited to contribute to their blood type. This can only be determined by either family studies or by performing molecular techniques. So by family studies, we mean if you know your blood type and you happen to know what your mother's blood type was and your father's blood type was, we may be able to deduce your genotype. Okay? So example, a person with the phenotype of A could have a genotype of AA, meaning they got an A allele from their mother and an A allele from their father, or they could be heterozygous, meaning they got an A allele from one parent and an O from the other. So we would need to do family studies to confirm. And we'll be doing some examples of family studies a little bit later in this presentation. Actually, it's coming up now. Okay, so here we see an example from our book. And when they're doing, they call these pedigree charts, which is kind of a funny name for them. But usually, you know, they have the mother as a circle and the father as a square, but this one's a little bit different. So here we see two parents, phenotype, their blood type here. One parent is A and the other parent is B. And here, you know, we could see the genotype, okay, which they must have deduced from previous family studies. And so we see this parent is AO, not AA, okay. And this parent is BO, not BB. And so basically we can do something called a Punnett square to predict the likelihood of different outcomes as far as their offspring with blood groups. And we're going to be doing that in a minute. I actually think it's pretty fun. And so what we can see here, you know, the end result is that when these two parents come together, there's a 1 in 4 chance they could have a child who's AB, a 1 in 4 chance of having a child who's A 1 in 4 chance who's having a child who's B, and a 1 in 4 chance of having a child who's O. So I like this example because sometimes people say, oh, well, my mother's A and my dad's B. How come I'm O? Uh-oh, right? Well, guess what? It can happen, okay? This other example here is interesting. We have a parent who's AB, and when we have someone who is type AB, this is one time where the phenotype is the same as the genotype. So we know that this person had to have gotten an A allele from one parent and a B from the other. That's the only way they could be type AB. And similarly with O, there's really only one choice for O as well. So when someone is type O, they have to have gotten an O allele from each one of their parents. They're homozygous here for O. And so when we do a Punnett square cross, which we'll be doing some in a moment, we could see here that interestingly, it's a 50-50 chance, okay? 50% chance, 2 out of 4, that they would have a child who's type A, and a 50% chance they would have a child who's type B. So here we see some Punnett squares that were done in our textbook, and maybe you've done one before in a biology class. I happen to think they're pretty fun. This was the last example that we looked at. So, you know, on the top of the Punnett square, we're going to put the alleles from one parent, or the A and B, and then we're going to put the other parent here. And so to do the Punnett square, then I'm going to take down the A from here, put it with the O, okay? Then when I move on to the square, I take this one down, and I still move the O, okay? We move on to the second row, the A comes down and the O, and the B comes down with O, and so we can see how there's that 50-50 chance of having A or B child. Let's look at some other ones. Okay, so let's look at this one. So here we have two parents who are both AB, and so in the first square, this A comes with this A, okay, so that would be a type A. We have the B come down with this A, okay. Here we have A and B, and here we have B and B. So we can see in this example that there's a 50% chance that a child would have the same blood type as the parents, AB, but then there's also 25% chance of having a type A child or 25% chance of having a type B child. So it's just a convenient way to summarize all the combinations of maternal and paternal alleles to figure out the likelihood of offspring. So let's get into ABO genetics a little bit. ABO typing can be used in cases where there is a question of paternity. And so in these instances, the ABO group of the mother, the infant, and the alleged father, meaning the person who the mother is claiming is the father, all would be tested. When we're doing this crossing with the Punnett squares, we have to trial each possible genotype for blood groups that may have more than one. So what does this mean? Let's say the alleged father in this case is type A. When we would do the Punnett square for him along with the mother, we would have to trial him as both AA and AO, because remember, someone who's type O could have either one of these two genotypes, and we don't necessarily know which one this person is, so we'd have to run it both ways. After doing the trial, if the child's blood type is not a possible outcome using the Punnett square, then the alleged father is excluded, meaning it can't be him. Okay, he is not the father. So here are some examples. And so in this example, we have a mother who's group A and an infant who's group B, and the alleged father is also group A. Okay, so the mother, okay, and the father. Okay, so here we would have to trial, do the trial twice, meaning we would have to do the trial with him as AA and then AO. Okay, and so here you can see AA. Oops, sorry, I made a mistake with this. It looks like the mother is AB. Okay, I'll have to fix that. Sorry guys. So there would be a B here. So the A comes down here with the A here, A and B. Okay, A and A and A and B. So when we trial the father as AA, we see that there basically is a 50% chance of a child that's A and a 50% chance of a child who's AB. So if we were just to trial him this way, we could say that, oh gee, there's no chance that he could be the father because the baby is type B. Okay, but we have to trial him a second time. Okay, so remember, we don't know if he is AA or AO. So, and once again, I apologize, the B is missing there, but here we have the mother who is AB, and we have the father. This time we're going to trial him as AO, and let's see what we get this time. So A comes down with A and B, A and O, and then B and O. And so when we look at the second example, we could see in this trial there's a 50% chance, okay, 2 out of 4, of a child being A. There's a 25% chance of an AB child, and then aha, there is a 25% chance of a B child, which is what the baby is. Okay, so in this case, you could see the importance of why we would need to trial him both ways. If we were just going to do it this way, we might say, oh, he's off the hook, but not really, okay, when we do them this way. And so I'm going to go back and correct the PowerPoint. I think when I edited it, something happened. So the mother is truly an AB, so she should be AB here, and I should have a B here. So I'll go back and fix that for you guys. So here we have a family pedigree chart, and this happens to be my family. And so here we see the square. This was my dad. Both my parents are deceased, but my dad was type A and my mom was type B. So when we start off, I didn't know the genotypes of them. So my parents only had 2 children. They had me, I'm type A, and they had my sister who was type AB. Okay, so basically, you know, when we look at this, there is one thing that we can deduce. My sister doesn't really give us much information because she got an A from my dad and she got a B from my mom, and so he could be AA or AO in that case, and she could be BB or BO in that case, because each one gave, you know, the A and the B. So that doesn't really tell us too much. However, when we look at me, okay, I'm type A, what we can say is, okay, well, I had to have gotten my A allele from my dad, okay, and I had to get an allele from my mom. Well, my mom was type B, okay, I did not get her B. So what this really tells me then, let me see if I can find my pen and try to write. What this means is my mom is really BO, okay, and she passed her O allele on to me. Okay, that's the only way it could work, because otherwise, you know, she would be, if she was BB, I would be AB as well, and I'm not. So I know my genotype has to be AO, and I had to have gotten my O allele from my mother because she couldn't give me the A. I got the A from my dad. So we really don't know what my dad is. Okay, so he could really be AA, or he could be AO. If, you know, I had another sibling, maybe it would rule it out, or if I knew what his parents' blood types were or his siblings' blood types were, I might be able to figure it out, but I don't. So at this point, I really can't say anything more about my dad. But, you know, hopefully you see how, you know, I definitely could tell what my mom was. Okay. All right, so I need to erase. Erase, erase, erase. All right, so let's do a trial together. What color should we use? I like the hot pink. Hopefully you like it too. So that's the eraser though, I don't want that. There's my pen. Sorry guys. Yeah, maybe it'll let me. I'm going to pause here for a minute. Figure this out and I'll be right back with you. Okay, so figured it out. All right, so we're going to look at example of BO and AO, and I think we may have looked at this one before, but just for practice, because you might have to do a Punnett square on a test. Okay, it's very important to know how to do it. So my first one, remember, A comes down, okay, with this B, okay, and here we're going to bring the A over with the O, okay, then the B comes down with the O, sorry about my writing, and then lastly each of the O's. And I like this example because you know, it's something that most people wouldn't expect. So what are our probabilities of a type A child with these two parents? 1 out of 4, or 25%. Okay. Same for B. 25%. Same for AB. 25%. And the most unusual one of all is the O, 25%. So this example I just find very interesting because sometimes, as I said before, students will say to me, oh, well, my mom is A and my dad is B, and how come I'm an O? And most people that don't know a lot about inheritance with ABO might say, oh, gee, what happened there? But it's absolutely possible. Okay, so you have to do your Punnett square to basically figure it out. Let's move next. All right. So it's really too bad that all of these drawings really carry over. Okay. So let's talk a little bit more about inheritance. And you may have learned about this in biology previously, but when we refer to a gene as recessive, this is a gene that must be inherited from both parents in order to be expressed. And so the best example I can give to you of a recessive gene has to do with hematology. If you think back to last semester when we were talking about sickle cell disease. And so we know a person with true sickle cell disease has a hemoglobin S from each parent in order to have the full-blown disease. Okay? When we're talking about our blood group system antigens, most of them are co-dominant. And what that means is one doesn't necessarily dominate over the other. And so basically, if you inherit an allele, it's expressed regardless. And we get equal expression of two different alleles. So if you think to the ABO system, which we just looked at, that's how we're able to have a group AB. Okay, so the A isn't dominant over the B or vice versa. They're both co-dominant, and that's how we're able to have both expressed. Dominant situation is when we have a gene that is expressed over another. Okay, and so there are genes, for instance, in the ABO system that do not express a detectable product, and those genes are called amorphic. And the example here is the O gene. So inheriting the O is basically nothing. Okay, it does not make a functional sugar to add to the chain of the antigen, and so we call it an amorph. So it's important to understand the difference between heterozygous and homozygous. So when a person inherits two identical alleles, we consider them homozygous. So AA, BB, and N. Basically, these are two of the same, okay? However, when we have a person who inherits two different alleles, we call that heterozygous. So AO, AB, MN, okay, they're not the same. Linkage is something else that is important with certain blood group systems that we're going to be talking about. And linkage occurs when two genes that are close to each other on a chromosome are inherited together. And when this happens, each set of linked genes is called a haplotype. And so here we see a pair of chromosomes, and we can see that on this chromosome, M and S are very close together. And on this one, N and S are very close together. And so these are examples of haplotypes because these genes are inherited together. So haplotypes tend to occur at a higher frequency than for unlinked genes. And so this results in something called linkage disequilibrium. Because they're so close to each other, they are inherited more readily together. Most blood group system genes are on autosomes, okay, except for one system called the XG system. So the XG genes are found on the X chromosome, which means if a father carries the XG allele, he will pass it to all of his daughters, okay, because he gives that one X to every daughter, but not to any of his sons because he does not give his X to a son. If the mother carries the XG allele and not the father, all of their children will have the XG because the mother gives an X gene to every child. She gives an X to her female offspring and an X to her male offspring as well. Okay, so next we're going to talk briefly about the concept of dosage. And this is something that we are absolutely going to revisit again later. It's a very important concept in blood banking. And dosage is important in certain blood group systems. So in some blood group systems, not ABO, but ones that we will study later, when someone is homozygous for an allele, okay, basically, you know, in my example, I'm using the M. Homozygous, they have an M and an M. It's almost as if they get a double dose of that M antigen, okay, on the red cells, when compared to individuals who are heterozygous for the M antigen. And so when we learn about these later, N is antithetical to M, and so it's possible to have both, okay? But when someone does inherits the M with the N, they have less of the M antigen on their red cells, because basically, think of it this way, the N is taking up some of that space, okay, that the M may have had all to itself, okay? So dosage is a variation in antigen expression when alleles are inherited homozygously. So I drew a very crude picture here, okay, and so I made a red blood cell. When we have a person who's homozygous for M, and I couldn't put it up here, so sorry, it would be going all the way around. You can see it's like they have a double shot of M. Look, M is like just basically embedded in the membrane all the way around. There's a lot of M, okay, because they only got the M. However, when they get the M and its antithetical N antigen together, we could see, look what happens. Okay, M, N, M, N, M, N. We could see that now M is not taking up every space that it could along the red cell membrane, because it has to share some of that space now with the N. So hopefully you would agree that there is absolutely more M antigen expressed on this cell than there would on this one when it has to share its spots with the N. And that's basically what dosage is saying. And we'll revisit that again later. So another important thing with dosage is we could look at antigrams of cells that we use in testing, and we could predict, for instance, which cells are going to react more strongly when someone has an antibody against a certain antigen. Okay, so here we see an antigram from some screening cells from our book. And using the MN example from the last slide, okay, if we look at the first cell, we could see M only. This is MM, okay, because they did not inherit the N antigen, which is its antithetical. So we're going to have much stronger representation of the M antigen on the first cell than we would on the second cell, where we see the M and the N are both expressed on this cell. Okay, so there's definitely more M on one than there is on two. How does this play out when we're doing our testing? So basically, let's say my patient has an antibody against M. Okay. When we test our patient with these screening cells, okay, and the patient happens to have this anti-M, they're going to react much more strongly with this first cell, where there's a lot more M antigen on the cell, than they are with the second cell, where M and N are both sharing the spots on the red cell. Okay, and so here we see the results of that. So the anti-M from our patient reacted 3+, okay, think about our 3+ agglutination, strong reaction with the first cell, compared to a 1+, a much weaker reaction with the second cell. And this is called the dosage effect. So this is simply because in the first example, they have a double dose of M, okay? So homozygous expression of some antigens will show stronger agglutination compared with antithetical antigens that are heterozygous. Okay, so if we were meeting face-to-face, I would ask you this question. I'm going to ask it, but I guess I'll have to answer it as well. Excuse me. So let's take a different example. Would cell 1 or cell 2 have stronger expression, and you don't know what this is yet, but this stands for Duffy, of the Duffy A antigen? Okay, so let's find Duffy. Here it is. Okay, so here's our Duffy blood group system. Maybe I will outline it. so everybody can see it. It's harder to do than you think. You have to use your mouse to use the pen. Okay. So here with our Duffy blood group system, we look at cell 1. We see it has Duffy A, but it also has Duffy B. Okay. When we look at cell 2, we see, aha, only Duffy A. Okay, so which one do you think would react more strongly? It's going to be cell 2, okay, because this has a double dose of the Duffy A antigen, whereas this cell is sharing it with its antithetical Duffy B. So hopefully that makes sense. We'll be doing more of these as we go, and I guess I better erase my things. Before I move on. Okay, so hopefully that makes sense, everybody. Okay. Okay, so the next slide has to do with theories of genetic interaction, and this states that the location of inherited genes in certain positions can affect the expression of the antigen. Okay, so what the heck does this mean? When we have alleles on the same chromosome, we call them cis to one another. Okay, so let's look at the second set. Here we see some antigens of the Rh blood group system. So we have the big D, which is the one that confers Rh positivity, but then we see we have a big C and a little e. And they are all located on the same chromosome. So we can say that D is cis to C and E. Okay? When we have alleles on opposite chromosomes, we call them trans. And so here we could see this individual also has big D, big C, and little e. However, were not inherited on the same chromosome, different chromosomes. So we will call these trans to each other. Okay, and we'll look at examples exactly of how certain blood group system antigens are affected later. But for now, hopefully you understand the difference between cis and trans. So next, we're going to briefly get into some studies of population genetics. And population genetics are used to determine the genotype or phenotype occurrence in a population, and we have to use two formulas. So first, we use a phenotype calculation, which enables us to find a unit of red cells that has certain antigen characteristics, meaning perhaps antigen negative. Okay, and so why would we want to do this? In blood banking, there are many people who have had transfusions in the past, and unfortunately for those people, oftentimes following transfusion, they can develop antibodies to antigens present on the donor cells that they lack. Okay, so these antibodies don't go away. Once an individual forms these antibodies, they remain with them, and they will impact the success of future transfusions for them. So let's say, for instance, as in our last example, we were talking about the Duffy system. Let's say we have a person who has it's going to be hard for me to write, anti-Duffy A, okay, from a previous transfusion, okay? When they come back for a transfusion, we want to find red cells that don't have Duffy A antigen on them, okay? Because if we transfuse them with red cells that have that Duffy A antigen, their antibody is going to say, hey, there's my target, form an immune system, and those red cells are going to be destroyed. So they're not going to be able to do the job that they're meant to do to increase that person's oxygen carrying capacity. The other piece of this is called the Hardy-Weinberg formula, and this is determines gene frequencies, okay, to produce a trait. So we're going to look at some of these next. So here's an example. So we have a patient with multiple antibodies. So they have an anti-C, an anti-E, and an anti-S. Sorry, that went on to the next line there. And so basically, when we know this patient has these 3 antibodies, we want to provide them with red cells that are negative, that lack these antigens. Okay? So what do we need to do? Okay, first of all, this is known from population data. We basically know the rate of positivity, how many people in the population in general have each one of these antigens. Okay, so we're going to go through and basically we're going to say what is known, and then using what is known, we're going to say, okay, well then what percent is negative, and then we have to convert it to a decimal to multiply. Okay, so basically, and this is data that is provided, okay, from research. We know 70% of the population has big C antigen. So if 70% of the population has big C, then we could deduce that 30% of the population does not. Okay, remember, we want to use the data that does not. Okay, so if we're going to convert 30% to a decimal, that's going to be 0.3. When we look to anti-E, or I'm sorry, to the big E antigen, we know that only 30% of the population has big E. So that means 70% of the population is negative for E antigen, and that would be 0.7, okay, if we convert to a decimal. Lastly, when we're looking at big S, we know 55% of the population has big S. That means 45% is negative, and convert to a decimal, it's 0.45. Okay, so remember, we're looking for units that lack each one of these, okay, so that our patient doesn't have a transfusion reaction. So next, what we do is we take each one of these decimals, 0.3 multiplied by 0.7 multiplied by 0.45, and what we end up with is 0.0945, and this is an estimation, so we could round this to about 0.1, which is the same as 10%. Okay, so this basically is telling us that 10% of the population would meet these needs, being negative for all 3, big C, big E, and big S. So therefore, 10% of the population is the same, if we convert to a fraction, as 10 over 100. And we can reduce that by getting rid of zeros to 1 to 10. Okay, so what this is telling us is we would need to screen 10 units to find one that is negative for C antigen, big E antigen, and S antigen. So about 1 in 10 units will be compatible. Okay, and so it's basically just giving us an idea about how many red cell units we would need to screen to meet the criteria. Okay, so let's clear. Okay, we're going to do one more example. Okay, so in this example, you have a patient with multiple antibodies. Okay, this time they have an antibody against Duffy A, JKB, and Big K. And we'll be learning more about these antigens and their blood group systems in an upcoming module. Now this time the patient needs 2 red cell units that lack these corresponding antigens. Okay, so once again, we're going to break it down. Excuse me. So we take what is known. Okay, so what is known, we know that 66% of the population has Duffy A. So therefore, we can say 34% is negative, and this converts to 0.34. When we look at JKB, we know 72% of the population has JKB, which means 28% is negative, so we're going to convert this to 0.28. Lastly, only 9% of the population has big K. So that means 91% of the population will be negative for this antigen, and that will convert to 0.91. So similar to last time, now we're going to take each one of these decimals and multiply them by each other. 0.34 times 0.28 times 0.91. We get 0.087, which would round to about 9% negative, 9 out of 100. Okay? Now, we need 2 units. Okay, so we need to do an additional step here. So to get 1 would be 9 out of 100, but we need 2. So if we cross multiply, we get 9x equal to 200, And if you divide that out, that's about 22. So basically what that's telling us is we would need to type about 22 units to find 2 that are compatible, negative for Duffy A, JKB, and big K antigens. Okay, so I'm going to move away from that and talk a little bit about molecular genetic applications. So if you move on to a 4-year program, you would be learning a lot more about molecular testing and probably doing some molecular testing as well. But molecular testing in blood banking has made great strides, and so it has quite a few applications now. And so earlier when we were doing our family studies trying to rule out an alleged father for paternity, etc., ABO typing that way doesn't always work. So sometimes genetic studies need to be done. So these are just some examples of different types of molecular tests that are done with regard to blood bank. So transplantation is a separate part that's really beyond blood bank. It's tissue typing is an area in and of itself, but just like we're studying red cell antigens, there are HLA, human leukocyte antigens, that are relevant to transplant. And so basically molecular tests are done to find the best match, okay? So with regard to blood banking and transfusion, we could do red cell typing in multiple transfused patients. We could determine genotypes, screen for antigen negative donor units more easily. So blood donor centers do this a lot based on molecular testing. We could do parental testing. And then, we'll talk more about this next one later. Donor testing, when you donate a unit of blood, our local blood center, the American Red Cross, screens for HIV, hepatitis B, hepatitis C, West Nile virus, and they are looking for the DNA of those viruses. So that's a molecular test. And then, of course, the relationship testing. So molecular testing is making great strides these days. Just going to briefly talk about PCR, polymerase chain reaction, because this is one of the molecular tests that really is a workhorse. And so with PCR, it rapidly and precisely multiplies specific sequences of DNA. And so here we see our DNA, double helix. And so firstly, they denature the DNA, meaning they unwind it. They add a primer that's going to attach to a specific part of the DNA, and then they replicate it and amplify it. Okay, and that's just really a simplification. And as I said, if you move on to a 4-year program, you will learn much more about molecular testing. So just wanted to briefly mention it. So that concludes Chapter 4. So when you have a moment, you should move on and do the Chapter 4 assignment and take the Chapter 4 quiz. I'll see you next time.